basically talked about the 3 sources of ATP for muscle contraction. We said that the first one, the most transient source, was from creatine phosphate, right? From creatine phosphate. And basically creatine phosphate was a storage place for that phosphate group, okay? In order to generate ATP, this enzyme, creatine kinase, was going to have to take the phosphate group from creatine phosphate, put it onto ADP, and that's going to give us, driving this reaction to the right, that's going to give us creatine and ATP. And we could use that ATP to contract muscle. This was transient. This was while those other 2 main metabolic processes were sort of warming up or getting started, okay? Now, when those processes got started, we no longer needed this mechanism. So we have this influx or this buildup of ATP, and so we start to drive this reaction back to the left. So ATP, right, and creatine is now going to give us, because this enzyme is reversible, is now going to give us creatine phosphate, which is sort of holding that phosphate again, holding that energy, and ADP. And this provided that source for the next time we needed this mechanism, right? So that phosphate is sitting on creatine, waiting to give its energy to the ADP for the next time we needed this transient source of energy, all right? So basically in the states when we have low energy or low ATP, this reaction is being driven to the right. If we have enough energy, right, those metabolic processes have taken over, this reaction is driven back to the left and it's sort of on tap, waiting for the next time we need it. Okay, and the enzyme that catalyzes this reversible reaction is creatine kinase. Does that answer your question? Yes? Any other questions on this or anything around here? Any other questions? Any other topics? Mm-hmm.
So is this creatine phosphate,
Mm-hmm.
is that present in every type of muscle fiber?
It is, in varying amounts. Mm-hmm. So this is always the first source of ATP. And then depending on what fiber type it is, it's going to kick into either oxidative phosphorylation or anaerobic glycolysis as its main source after this is depleted. Any other questions? Any other topics?
Can you explain what funny channels are?
Funny channels, sure. So we talked about funny channels last time, right? Now, these are channels that are on your pacemaker cells, okay, your pacemaker cells. And their main job is to allow the movement of sodium into the cell. Okay, so that's their job. If we wanted to polarize this cell, the first thing we do is we close a potassium channel, right? We open up this funny channel, and because we've closed potassium channels and opened this channel, which is allowing sodium into the cell, we start that depolarizing of the cell, okay? Now this initial process is going to trigger a slow calcium channel to also open, and that's going to be the initial, so there's the slow depolarization, then the initial depolarization, and then the faster calcium channels is responsible for the rapid depolarization. So basically a funny channel is basically starting the depolarizing of the cell by bringing more sodium in, okay, and that's going to make inside the cell more positive, and it's going to also trigger the opening of your slow calcium channels. All right, did that answer your question? It's going to start helping that pacemaker cell depolarize. And remember, these can generate action potential spontaneously. So in the absence of any other external stimulus, the membrane permeability is going to vary here. These funny channels will open up spontaneously, and that will start the depolarizing of the cell. This is specifically on your pacemaker cell. Now, this mechanism is where we talked about how the sympathetic system influenced these cells. Right? It's going to keep them open for longer. So it's going to make it very easy for the cell to be depolarized. All right? And hence it's going to increase your heart rate by doing so. Any other questions? Any other topics? Sure. So the muscle spindle. Talk about the muscle spindle. Two. All right, so the muscle spindle was basically that receptor in the center of the muscle that was responsible for detecting the change in length. Okay, so if this muscle stretched beyond its length, if it was stretched, the natural reaction that the body wants to have is to contract that muscle in a protective measure to prevent overextension or overexertion of this muscle. Okay. Now, they're the extrafusal fibers. These are outside of the muscle spindle. These are your normal contractile cells, right? They're innervated by an alpha motor neuron, which is a faster neuron, has a larger diameter. And the inner muscles, they're also muscle fibers. The intrafusal fibers are also muscle fibers. They contract, they have your sarcomeres, right? They are just modified, and they're modified in that they are, one, they are associated with these two nerve endings we talked about. Two, they are innervated by a different motor neuron. So they're innervated by a gamma motor neuron, and then they are intertwined or interacting with these two nerve endings. The annulospiral nerve ending, which is a type 1A afferent fiber, and the flower spray nerve ending, which is a type 2 afferent fiber. Now, these two fibers, these two afferent fibers means that they're going to pick up any changes that's going on in this muscle, and they're going to send that signal to the central nervous system. So if you stretch a muscle, the annulospiral ending and the flower spiral ending, they're nerve endings. So they're sensory. They're picking up information about what's happening. They detect that change in length. They send a signal to the central nervous system in the language of increasing the action potential firing rate. So that's the language that the central nervous system will understand. All right, so you increase the action potential firing rate, that sends a signal to the central nervous system, and the central nervous system wants to send back a signal on efferent fibers to allow that muscle to contract or to shorten, again, in a protective reflex. So then that signal comes back down on your gamma motor neuron, an efferent fiber, and your alpha motor neuron, also an efferent fiber. All right, the trouble here is the gamma motor neurons are bigger, they have a faster conduction velocity. So basically the extrafusal fibers will contract first, and then the intrafusal fibers are gonna contract to meet that. All right, and they have to adjust, they have to readjust to meet the extrafusal fibers so that they can continue to detect any further changes in the length of this muscle. Question?
Just to clarify, the alpha motor units are
Correct. The alphas are going to the extrafusal fibers, and the gammas are going to the intrafusal fibers. Any questions, guys? Any questions on this? I hope that answered your question.
Which one is bigger, did you say?
So the fibers aren't necessarily bigger, but the axon diameter, so that nerve, is bigger. The alpha motor neurons, which innervate the extrafusal fibers, is bigger. So the axon diameter is bigger for the alpha motor neuron, as opposed to the gamma. Any other topics? Any other questions on this? No? That means everyone's gonna get 100? I hope so. Yeah, if you have any questions between oh,
Back more on the channel pressures and like how it passes.
sure. So the cardiac cycle, basically, we can go over the cardiac cycle. All right, so let's review the cardiac cycle. And we'll start by going through each of the phases, right? So the first phase is called ventricular filling. This is where your ventricle is receiving blood, okay? Your AV valve is open and the pressure where has to be higher? In the atria or the ventricle? The atria. So we said that blood wants to go from a place of high pressure to a place of low pressure. Okay, so the opening and closing of valves is not an active process. It's basically passive. When these pressures are in the right amount, in the right areas, a valve will just normally open up. If pressure is higher in the atria than it is in the ventricle, the AV valves open up. Okay, blood wants to go from the atria down into the ventricle. This is your filling phase, right? Now, as blood fills into that ventricle, the pressure begins to rise. And as the pressure rises, when it gets higher, when the pressure in the ventricle is now higher than that in the atria, this blood no longer wants to go here, right? Pressure here is now too high. And so that AV valve closes. Right? The AV valve closes when the pressure in the ventricle rises beyond the pressure that is in the atria, and we lose our pressure gradient. We no longer have a pressure gradient. Okay? Now the pressure in the ventricle is going to continue to rise because this ventricle is now contracting. But it's not high enough yet. It's not high enough yet to open up the semilunar valves. Right? As we just said, it needs to be higher than the pressure in the aorta to open up the semilunar valves. So in the next phase, this is isovolumetric contraction, the pressure in the chamber is rising, right? It's higher than in the atria, but it's lower than in the aorta or the vessel. And so it's not enough to open either of those valves, okay? So in isovolumetric ventricular contraction, the pressure rises and the volume remains the same, simply because both those valves are closed. In phase 3, this is your ventricular ejection phase. This is where the pressure in the ventricle has now overcome the pressure in the vessel, and so blood can be ejected from the ventricle now because those semilunar valves can open up. All right, we have higher pressure in the ventricle than we do in the vessel. And so that semilunar valve opens up and blood is ejected. Now, as blood is leaving that ventricle, right, pressure is dropping. As we're squeezing blood out of the ventricle, the pressure in that chamber is dropping. And it's going to drop and drop and drop and drop as blood leaves and leaves and leaves until it is now lower than the pressure in the vessel, and we lose our pressure gradient once again. When we lose our pressure gradient, the semilunar valves are going to close again. Okay? So the semilunar valves open at the beginning of phase 3, and then they close at the end of phase 3 because we've lost the pressure gradient. In phase 4, we have isovolumetric ventricular relaxation. The pressure in the ventricle now is dropping, but it's not low enough where it's lower than the pressure in the atria. So we don't have a pressure gradient just yet. All right? It's not low enough to where it's lower than the pressure in the atria. In order for filling to start again, that ventricular pressure has to go below the pressure that is in the atria. All right? Otherwise, we don't have a gradient anymore. Okay, so until that pressure drops lower than what's in the atria so that we can have our gradient again, we don't have filling. When that happens, right, when that pressure drops lower than what's in the atria, we start over again with the phase 1 when those AV valves open up again. Okay, so basically the pressure in each of the two chambers is dictating which valves are open, which valves are closed, and where the blood is flowing. There needs to be a pressure gradient. The pressure is highest in the atria and lower in the chamber, blood will go to the, highest in the atria, lower in the ventricle, blood will go to the ventricle. If the pressure is high in the ventricle and low in the vessel, blood will go into the vessel. If the pressure is low in the ventricle, right? If the pressure in the vessel is higher than what's in the ventricle, blood will not want to go into the vessel, right? That semilunar valve will not open. And likewise for the ventricles and the atria. All right, so we have to have a pressure gradient for blood to move and the valves to open. Any questions on that? Any questions? Did that answer your question? Yeah? Any other? Mm-hmm. Sure, the aortic pressure. That was in this packet. All right, so the aortic pressures, now we want to understand where we're talking about here. We're talking about the main valve or the main vessel that leaves the left ventricle. This is going to feed all of the blood in your systemic arteries. This is going to give you a carotid artery, subclavian, all those distal arteries comes from that big vessel called the aorta, right? Now, when the semilunar valves are open during systole, the pressure in this valve, or the pressure in this vessel, sorry, is going to be high. Start right here, it's going to be high, this is systole, and then it's going to drop off as that blood goes out into the systemic arteries, okay? When that pressure is its highest, we call that systolic pressure. So the highest amount of pressure that is in the aorta is your systolic pressure, okay? Now let's look at diastole. At diastole, the pressure in this valve is pretty low, it's decreased. Okay, the semilunar valves are closed, blood is sort of going away, but there's no additional blood coming into the aorta, and so that pressure is decreased. When it gets to its lowest that it can be, that is your diastolic pressure. So the highest pressure in the aorta is your systolic, that's your top number. Your lowest pressure in the aorta is your diastolic, that's your bottom number in terms of your blood pressure. Now, if you averaged the amount of pressure in this vessel at any time in the cardiac cycle, right? If you took it in diastole, took it in systole, and you average that out, that's called your mean arterial pressure, right? So another way to say that is mean arterial pressure is the average amount of pressure that is in your aorta at any time in the cardiac cycle. Okay, any questions on that? Did that answer your question? Right, so the main takeaway message with aortic pressure is during diastole, when you close that valve, the pressure drops all the way, that lowest number is diastolic pressure. During systole, the semilunar valves open, the pressure rises, that highest number is your systolic pressure. If you average those out, that's your MAP. Okay. Any other questions? Mm-hmm.
Can you go through the conduction of system of the heart, like the bundle of tests with the cardiac?
Okay, yep, sure. So the conduction system of the heart, that was in this one. All right, so the conduction system of the heart was basically how electricity moves through the heart, okay? Now we know that the action potential or the impulse is going to start in what type of cell? A pacemaker cell, right? A pacemaker cell. What are the two congregations of pacemaker cells that we have called? Sinoatrial node and the atrioventricular node. So the SA node and the AV node are basically our two types of pacemaker cells. So the first impulse is going to start at the SA node. Okay, now the SA node is going to fire, and it's going to not only excite the entire atria and the left atria, so it's in the right atrium, but it's going to excite this entire chamber as well as the left atrium. That impulse spreads across both atria. Okay, and that allows them to contract. Okay, then the signal goes to the atrioventricular node. Now the AV node is in between the right ventricle and the right atria. So in between this area here, right? And that sort of is acting like a delay. That is going to delay the impulse from getting down to the ventricle prematurely. We want both of the atria to contract before we get that impulse going to the ventricular tissue. Right? So now the impulse goes from the AV node down to the bundle of His. The bundle of His is basically in between the septum that separates both sides of your heart. So the septum here, separating left and right ventricle. In between that area is where we have your bundle of His. So we're not exactly exciting the entire ventricle just yet. Okay, we're just trying to get the impulse down to the bottom of the heart. Okay, so that impulse goes from the AV node through the bundle of His, and then it's going to split off into your left and right bundle branches. Right, you have your bundle branches going up to the right and your bundle branches going up to the left. Okay, it splits off, and then it's going to relay the signal to your Purkinje fibers. Your Purkinje fibers are going back up towards your valves. And so the heart is going to contract from the atria, and then the ventricle contract upwards. So up to the bottom of the atria, to the base of the heart, and then the ventricles contract upwards. Okay, and that is the relay of the electrical impulse. And if we look at this image, so this image here, or this sort of representation here, we can see that the atria contract, then the ventricles contract. And the ventricles are contracting from the bottom upwards. All right, so we have to contract the atria, then get that impulse down to the base of the heart, and then it envelopes both sides of the ventricle.